So, welcome everybody. Today's talk will be about molecular machines, a bit at least, and we will focus on some subclass of molecular machines, those ones which are in interaction with the cytoskeleton. So this is also the last talk for today, and I will make no more movies, will put it in, also from colleagues who also animators who made some fantastic illustrations which I cannot do, which you have to use a lot of knowledge of 3D software. We will also use it a little bit, to get something of the excitement about molecular machines. Now, first of all, what are molecular machines at all? There is, I think, a quite useful definition by Jarmusch and colleagues. Published this paper in 2004, this review. Molecular machines are tiny energy conversion devices on the molecular size scale. Whether naturally occurring or synthetic, these machines are generally more efficient than their macro-scale counterparts. Superiority can be, of course, in different ways. Superiority can be in terms of the kind of efficiency, in terms of energy usage, but very often molecular machines also can act reversibly, like an electrical motor, which also when you reverse it is becoming an electrical generator. Many of these molecular machines can be also used in a reversible manner, but not only. So then I was wondering, molecular machines is I think a word or a term that is very often used by all kind of people. Someone is studying a simple enzyme and it's calling it a molecular machine. So I was also looking a little bit for a bit more useful classification of what a molecular machine is and how one can understand them. I found then. So this review from Watson and Cockcroft gave some, I think, quite useful framework by asking 4 questions and whose answers are determining what kind of machine we actually do have. The first question is, when is a machine, does it exhibit discrete kinetically trapped states? So completely, or not completely, but different conformational states which correspond to different steps of its overall function. Think about a motor and the piston in the cylinder, and adopting different kind of positions during its action. So when this question is answered with no, it is not a machine. It may be, for example, a simple enzyme that is catalyzing some reaction with some transition state, and that's just it, and is making causing products and substrates to be set into an equilibrium. It could be also something that has to do with open channels like our porins. Yes, they are certainly not a molecular machine. Yes, or something like our potassium channel. It is open, but it does not have a discrete state at all. Actually, it works without activation barrier. Folder mass and some complexes can be something like looking a bit complex, but not a machine. And random walkers which walk in some arbitrary direction or just exhibit stochastic behavior. However, when the question is yes, we are talking about machine-like systems. Second question, does it perform work such as moving a particle energetically uphill? When the answer is no, it adopts different conformational states, kinetically trapped states. For example, you could argue that bacteriorhodopsin, this kind of light-driven ion pump, that adopts discrete states. Yes, indeed. Actually, it's also moving a particle, in this case a proton, energetically uphill. But they could be just also simple transporters, bistable channels, or just very simple chemical analog of a one-shot directional walker. So when it's moving something energy uphill, it is clearly not machine-like, it's a real machine. Does it continuously perform work without external switching of conditions? When the answer is no, then it's actually a machine that is not somehow driven by the external conditions. It works continuously. It's a non-autonomous machine. When it's dependent on some external conditions, then actually it is a machine that can be autonomous. Does it continuously perform work by transducing energy from a chemical fuel like ATP or some proton gradient force photons? Remember bacteriodopsin or the photoreaction center, or non-fluctuating electromagnetic fields? Yes, and there are some examples of those ones given. So in the same review, they give also some examples, of what chemists, supramolecular chemists, can do nowadays. Yes, supramolecular chemists are now very interested in these kind of systems. They often use here, for example, systems like these rotaxanes, which can adopt different positions on a linear template depending on the switching conditions on some subreactions occurring. For example, in this kind of relatively complex machine that was supposed to be a simple peptide synthesizer, because you see here one building block is here attached to the rotaxane. It can move in a directional way when it finds the next building block, here block protected, some of course the environment has to be properly, it can make the next step and moves it on and on and on. Another example of our chemical friends are this kind of a redox-driven pump, where you have a single position, this one here, that can be reduced, then it becomes electrically neutral, so the positively charged rotaxane-like compound can bind to it, and it's trapped here in this position. When you then reoxidize it, it's getting positively charged again. Here's actually a block that this rotaxane is not moving on, When you oxidize it positively, there's electric repulsion happening here between the rotaxane and the linear template, driving it unidirectionally forward. Something like, you could call it a redox-driven pump, At least it's some chemical analog of this. And I also recommend to you are mostly chemists, also to look what was the Nobel Prize of Chemistry for. In this case, it was not for biochemistry, which is very good. It's also for something different, to Feringa in Netherlands, Stoddard and Sauvage, about making very nice examples of molecular machines. So when being in life sciences, molecular machines, the usual suspects, which you should have already learned in some context. I show you just the movies of those ones. The ATP synthase, that is responsible for synthesizing ATP from ADP.
Life requires energy.
Yeah, it requires energy. I switch this off. I hope that you get me still. So it's using it generates ATP, and it makes this kind of high energy bonds, acidic anhydride bond between this phosphate and the ADP, and this is clearly a strong uphill reaction. So the ATP synthase is a reversible enzyme that can work in both directions. It can hydrolyze ATP to ADP and phosphate and make some mechanical work and pump some protons, or it can go the other way around. It can synthesize ATP from these two EDUCs to ADP to ATP by using the flow of protons through its complex. So this is a very simple machine. Very simple means we have here a rotamer that can rotate. It's a membrane protein comprising of 10 to 15 different C subunits. The number is variable. So you have here the catalytic subunits, where the ATP is synthesized in the interfaces between the alpha and the beta subunits, and you have the stalk region that is anchoring the catalytic part here with the transmembrane part, where you have the proton flow. Now we have a gradient of protons here shown in yellow. So these protons now occupy the protonation sites here, and now the thing starts to rotate, because it rotates by releasing every time a proton here from this position. It's getting occupied here, rotates by almost 360 degrees, and then release a proton. And this rotation movement is transmitted into the movement of this rotor stalk here inside the catalytic site. Here we see some detail. Here's the A subunit. Here's the B subunit. Here's the rear channel where the proton comes in and occupies the first position of the aspartic acid residue on the C channel. Remember, the aspartic acid residue is here just in the midst of the membrane. And here's the front channel when it comes into contact with protein aspartate, it can release the proton to the cytosol. And now we have here this rotating central stalk. And this is causing a mechanical movement of the catalytic domains all the time over. And now we see the top view of the central stalk. And you see these interfaces are unequal. There's alpha-beta interface, alpha-beta interface, alpha-beta interface. But they are different at each time point. One is capable of binding a new molecule ADP and phosphate. Then there's a rotation happening. It changes to a state where actually now the formation of the bond can happen, and releasing then finally then the ATP. So that is a rotatory catalytic mechanism of ATP synthesis, the very basic part of all life that we do know, because most organisms have an ATP synthase in their plasma membrane. We have it, of course, in the mitochondrion. We also do know where the energy, what it's required for the mechanical energy. The mechanical energy is mostly required for changing at the end the conformation of the alpha-beta subunit in such a way that the product can be released. It's interestingly not required for forming the bond between the ADP and the phosphate, because there will be is energy expedited to remove all water from the catalytic site itself. So this is a water-free environment, because water would be, of course, a substrate for the counter reaction, the hydrolysis of ATP to ADP and phosphate. And that's, how that looks like a bit more in complex terms. When we are in a mitochondrion, they form also some complexes. Here you see some dimeric ATP synthesis causing here a kink in the plasma membrane. These are some of the suprastructural requirements the ATP synthase is also doing for forming, for example, the crystal in our mitochondria. So that was example number 1 from the Stewart lab and from Daniela Stock, who is in Australia and works quite a bit as in structural biology. So let's go for the second system, the bacterial flagellar motor. Also a classic system, an absolute classic system. We have it in all bacteria who have a flagellum and can move. We see a typical bacterium, 1-2 micrometer length. We see here some of these kind of pili-like structures, but these pili-like structures, you see here now they are in the movement. You see these bacteria, they swim through the liquid. Just imagine the viscosity. When you would be the size of a bacterium, it would be like you would swim through an agar gel, because it's highly viscous. So it's somehow propelling through the gel. So we see here the size relationship between our ATP synthase we have talked before and the G protein coupled receptor like the rhodopsin, which we have seen as well. But this is the size of the flagellar motor. It's much, much larger. Maybe I can go back here for this movie. It's already a bit fast. Made here some jump, I'm afraid. So that was ATP synthase. That is the size of the flagellar motor. The super huge complex. You see that in the microscope, yes. We have here again a rotary part, yes, here made up by these free F subunits. And we have here the free G subunit and the motor subunits here, which are part of the inner membrane. That one here is of the outer membrane, free M, free N, and also proteins like QY, which are associating with this. So we see here the rotor, super large rotor. We see here the inner membrane. We see here the outer membrane. We see here the shaft of that one. We need a stator. Every kind of rotatory motor needs a stator. So you see, this is actually stabilizing the structure. And the simple is the idea is very simple. A proton is coming through this kind of stator-like structures and gets to the cytosol all the time. And that is actually causing the rotatory movement in a very similar way. So we have here a gradient between protons in the extracellular milieu and here protons in the intracellular milieu. And this proton gradient, the flow of protons through this kind of parts of the between the stator and the rotatory fragment drives the whole rotor and the flagellum that is attached to it forward, so that you get this kind of fancy flagellar movement happening. So that's really exciting, but I will talk today a bit more in detail about something that is more closer to humans than flagellar motors. So I will talk about motor proteins that have to do with our cytoskeleton, because it's something for our basic life, So that's something, I don't know whether you learned it in this kind of biochemistry 2 or 3 still, maybe yes or no. In case yes, then you get it redundant. In case no, then you don't get it redundant. Just to remember to you, so we are a multicellular organism. So we have, of course, very differently shaped cells. And the cytoskeleton actually is a very complex network made of protein filaments. So not monomeric proteins, but really filaments, which are formed inside the cell. So these filaments, they organize the distribution of the cellular contents. For example, the ribosomes, where the mitochondria are, the nucleus, where it's more to the membrane or dividing or whatsoever. They transport subcellular particles, which is very interesting. Think about an axon of your neuron transporting some secretory vesicle along this very long distances you sometimes have, from centimeters to almost a meter. Shape formation and change of the cell. Yes, cells have shape and they can sometimes change their shape. Also, bacteria have shapes. So you will see we also have cytoskeletons in prokaryotic organisms. And of course, this kind of cytoskeleton is also required for cell motion. And that's actually also the part where actually in the last 3 points, transport, shape formation and change and cell motion, our motor proteins do come in. Just, to get you back, about cytoskeleton, we have 3 major components: microtubuli, microfilaments, and intermediate filaments. The microtubuli are consisting of protofilaments, which are made by heterodimers. These heterodimers are made of alpha tubulin and beta tubulin. So that's a basic block, alpha beta tubulins, heterodimers. In the case of the microfilaments, these are actin fibers. So it's called F-actin, chains of filamentous actin, F-actin, and these are made from globular actin monomers, G-actin. So there's something of an equilibrium between G-actin and F-actin. And at the end, we have something like the intermediate filaments. The most famous one is your hair on top of your head or some other places, but we have this kind of intermediate filaments not only outside our body, they are also forming a network inside the cells and to give the cells also form and stability. And they are not dynamic as the first 2 ones. They are more or less static in terms of their kind of architecture and polymerization state. So I don't read now the whole slide. You see there are a lot of different functions here now. We come to some of the functions later. So about the intermediate filaments, maybe because they are the simplest one, I make some notion. Yes, they are exactly built like keratin. Actually keratin, the monomer of our hair, is also the same protein family as intermediate families. It also shares sequence similarity with other intermediate filaments. But we have quite a lot of different intermediate family filament proteins. For example, here we have, besides the keratins, we have nuclear lamins. They are in charge, of giving the nucleus this very nice rounded shape that we do know from microscopy. We have neurofilament proteins, which are also very important, to make this very nice dendritic structures that our neurons are making. So they also stabilize this. And vimentin is also occurring, for example, in microvilli-like structures, as we will see later in context with actin. So come to something that is now much more interesting than intermediate filaments. Microtubuli. I find that fascinating. The molecule is relatively simple. It's binding, as usual, some GDP. Yes, it's also not too unusual, so you can already think it's probably one of the domains is a GDPase domain, as we have seen it with P21RAS. Actually, it's the same protein family, more or less. These kind of alpha-beta tubulin heterodimers, they form spontaneously. They can form spontaneously these protofilaments, which are made up by 13 rows here, 13 protofilaments, yes, making this tubule, 25 nanometer. You can nicely see it in the electron microscope, seen here, negatively stained. And of course, as you can imagine, this is polar. There's one end, that is plus and the other one is minus. These ends are different. So what's amazing about these tubulin fibers, this microtubuli? They're extremely stiff. So one of the characteristics of stiffness is the so-called persistence length. That is giving us some idea, how over what distance we keep, let's say, our rod-like structure of anything or stable structure. To have a millimeter long microtubuli, we have to have thousands and thousands, 10,000s of these kind of heterodimers attached to these protofilaments. So this is amazingly stiff, and you see this is because of this architecture this kind of microtubuli takes. They are hollow filaments. Inside, of course, filled with water and whatsoever. They are formed tubus, but they are stiff. That's really efficient. So they have a positive and a negative end. You see that here's alpha beta is coming in. So at the plus end, this one end here, this is usually the site of rapid polymerization. So when a microtubule changes its length, it's usually growing at the plus side. It can also shrink from the plus side, of course. But this is a site where it's actually rapidly growing. The other side, however, the so-called minus side, can only slowly polymerize. It's also the preferred size for deep polymerization. So you can imagine you can have it one and the same microtubulus grows at the plus side and shrinkage at the minus side, which causes then, of course, the microtubulus moves virtually through the cell and makes some movement already on itself. So to get a bit of this idea of this kind of microtubuli, please remember this is a process where you have monomeric, or not let's say globular alpha beta heterodimers here in solution. In order to form a filament, they have somehow to nucleate. This is the same process like we have it in protein crystallization or in the formation of some aggregates like in Alzheimer's disease, So we have to have somehow to meet a lot of these kind of alpha beta tubulins together, 8, 9, 10, that they form a nucleus. And the nucleus can then form the starting part of this kind of tubus-like structures. And then you can have elongation, not at the minus side, but at the plus side. And when you're in steady state, you have an equilibrium between addition of new alpha beta heterodimers and dissociation of alpha beta heterodimers, and usually from the plus end, because the minus end is very often also blocked from depolymerization by interaction with microtubuli associated proteins. So the whole process here, nucleation, elongation, steady state, stability of plus and minus ends, is of course subject to interaction with many, many other microtubuli associated proteins. So what is causing stability and growth of microtubuli? Microtubuli and tubulins belong to the G proteins. You have learned that the G proteins undergo conformational change upon exchange of GDP with GDP and upon hydrolysis of GDP to GDP and phosphate. So in the GTP bound form, the alpha beta heterodimer associates and forms a stable cap here, So this is growing. As soon as here GDP is bound. Behind it, yes, it's already hydrolyzed to GDP. So because of its intrinsic time trigger point, after some time it hydrolyzes it. But it doesn't matter, as soon as we have enough here at the plus side of GTP bound alpha beta heterodimers. So here is growth happening. However, when there is no new alpha beta heterodimer associating with GTP to this GTP cap-like structure, and it hydrolyzes because nothing is added, everything falls apart. It's destabilized. So it starts to shrink then in the case when you have here only GDP bound form. So that's actually one side of control. You speed up, for example, the rate of hydrolysis. The thing will also start to blow up itself. One of these things is, it's this exchangeable GTP, clearly, there's a kind of conformational trigger by the association of the GTP-bound tubulin dimer to the protofilament. It's causing some kind of conformational chain effect also on the GTP side. It destabilizes GTP there. It also causes a local curvature because the conformation change we have in the small subunit. And what is most interesting for us as chemists, or as also, let's say, people who are also interested in using this kind of effects, is there are a lot of toxins existing which are just targeting tubuli, microtubuli. So here you see an overview of the most famous ones, like the colchicins, which destabilize the plus end. You can imagine plus end destabilization, will cause an, let's say, a lysis of all this kind of tubuli. So, for example, cell division is automatically inhibited, yes, because cell division requires on the division of the mitotic reaction. The mitotic reaction requires tubuli because they are part of the spindle apparatus. So destabilization microtubuli is deadly. Cells will die from this. So these are all cytostatics. All of these compounds are toxic. Another very famous compound is Taxol. Here you see the commercial name, paclitaxel, It's also cytotoxic. With this one, you can also treat fast-growing lymphomas or some cancer variants. Also by changing the microtubule. In this case, however, the deadly point is by stabilization of the microtubule. Because what will happen then? When you stabilize the microtubule, you will consume all three alpha-beta heterodimers which are in solution. Everything will be then polymerized and no reaction will further occur anymore. There will be also no cell division reaction as well because there is no dynamics anymore apparent. So there are a lot of different compounds that have been synthesized and developed by medical chemistry against this process. Let's make a look on the alpha-beta heterodimer. Actually, that was interesting. It was structurally characterized the first time by two-dimensional crystallization electron crystallography. Very interestingly, yes. One of the few examples where electron crystallography was good for something. So we see here a complex between the GTP-bound alpha subunit and the GDP-bound beta subunit. But here, this one is important, the GTP-bound form. The topology is here like we have it with our G proteins. You see here the phosphates. We also have a P loop that is associated with the triphosphate group. Very strangely, in our microtubulins, in the tubulins, we don't have this conserved motif, this GKS motif that is otherwise found in almost all ATPases or GTPases. It's different here for some reason because hydrolysis has also to be slow. It has to be on a slow time scale, and actually association is much more important for this kind of proteins. So one of the things to regulate activity of these tubulin or of tubulins is association with tubulin-associated proteins. One of these proteins is stadmin. You see this here. So you see this is really something where you can predict when this stadmin is not associated to this alpha-beta heterodimers, it will not form a long alpha helical structure at all. It will fall probably in part and will be probably also in major regions unstructured at all. So the stadmin is a very nice example that association with other proteins can form structure for a given protein. In this case, it's quite interesting because here we have colchicine bound to the alpha-beta heterodimer, and the colchicine is actually causing a destabilizing reaction of the plus ends, and it's also changing the association here. And by this, it's actually very nicely also associated in this resting form, not being part of tubulin with the stadmin. So that is where actually stadmin can interact with. Here you have the overview of the roles we have for our microtubuli. Yes, some know that, of course, from biology, for example, in mitosis, it's very important. I mentioned already the spindle apparatus, where you have the separation of the chromosomes. And here, these are the fibers. These are microtubules where the chromosomes are attached to it. And here's a spindle pole, which is a densely packed microtubule structure. Very, very important. Here also in the interface, we have here for the formation of the cell shape, cilia are can be formed by our tubulins, cilia and flagellum. They are membrane, of course, surrounded, not like the bacterial flagellum that we have seen before. And here in a nerve cell, they form the central core, the central, let's say, road, where the minus end is close to the cell body, and the plus end is actually at the end of the axon, for example, close to the synaptic cleft. And interestingly, this is acting as a road because you can transport along this road cargo. One of the major roles we have for tubulins, for microtubuli, is forming roads inside the cell along which you can, here, like this walker here upside, unidirectional walker. For example, a walker that walks from the minus end to the plus end and transports something. The black and yellow, the black and gray ones is a motor protein, followed by an adapter, and the cargo is here the round, in this case, vesicle. But it could be also a protein complex whatsoever that can be transported given a suitable adapter. So you see that kind of idea here as well. So the kinesins are in charge of the minus end to plus transport, whereas the dyneins, these motor proteins, related but not so similar with the kinesins, do exactly the opposite direction, from the plus ends to the minus ends. And the common thing is we have an adapter, so one motor protein, one adapter, or several adapters, and many, many different kind of charges, cargos, which you can actually move to. So let's make a look to the kinesin motor domain. Kinesin motor domain is also something that is working with an ADPase-like system. Here we have in this case, it's the ADP structure. It's an ADPase domain. We have a P loop as usual. Yes, that is not so different. But we have also something that we see a bit better here. Also something attached to it at the C-terminus. Something like a lever, yes. A helical neck region, yes. So this is a dimer. This is a functional part. Here we have now something where this helix is dimerized, makes a dimer, and we have now these 2 feet now attached to this helical neck. That is actually the functional unit. That one here, the monomeric kinesin, of course, it can do some conformational change, but it will not be able to make a unidirectional walker with a monomeric kinesin. So the idea for the kinesins is that we have here our neck region, our ATP. We have again our P loop, which we always have, because it's associated to the phosphate. And we have, as we have seen it with the G proteins, our switch regions, which change the conformation upon hydrolysis. So what is happening here in that case? When hydrolysis happens, it is transmitted first to the switch regions, but they also have an effect on this relay helix. So you see that from left to right, the helix changes its position relative to the main body of the domain. And by the change of this relay helix, the neck that is associated with the relay helix gets being released from the association. Here we are in the ATP bound form. Neck linker is associated with the relay helix. Here we are in the ADP bound form. It's now released from that. That is interesting. The motor protein is relatively simple. We have at least 14 different orthologs in our genome. Depends a bit, more or less. Some organize more, some less. Let's see whether this movie will work. So that's a simple movie, yes, So we bind now in the ADP bound form to the microtubule. There we bind. we exchange to ATP. Now actually the neck region gets being associated to the relay helix and it is pushing it forward now. And the other one, you know, is now also exchanging. Here is hydrolysis happening, and by the hydrolysis happening here, it gets loose again, and is making this movement. So that's a very simple motor, actually. Yes, just by having 2 feet, yes, you know, which change the kind of association, you know, between this relay helix and the neck region. Cool. I find this cool. I like motor proteins. We have, of course, also a more fancy movie here from. I found this from Discovery Science. Let's make a look here. You know, everything looks here, of course, more flashy. Let's make. So you see, I like these animations. You know, that looks like a mitochondrion is being transported now. That could also be a virus. I don't know what this is. You know, maybe something nasty. Here are our kinesins. You know, attached. In this case, probably a vesicle. Yes, you know, attached to it. You know, and you see there even also several kinesin dimers associated now with this one. We are the kind of adapter to the kind of cargo, and they move slowly here ahead and ahead. And we have seen the mechanism, the unidirectional association, dissociation of the neck region from the relay helix, and that one is being controlled by the switch regions. Cool. I like these movies. There are even more, yes, but what is happening here, you know? Oh, something is moving. Oh, I like this kind of animations. Cool. Absolutely cool. Think about coronavirus. Walks up now, maybe you get infected. You get it into your or into your nose, yes, where you actually have the receptors for different kind of smells, yes, you know, they incorporate it, but they have to get to the neural bodies at the end in your reptile brain. Yes, it takes a few centimeters to go there, so they attach to an adapter, and that's being transported along this fantastic tubulin into your brain. Yes, so cool. Yes, you know, transport is fantastic. I just mentioned a lot of associated proteins with microtubuli. You see the kinesins have been actually the mobile class of them, but they're also more or less static ones. You see this here as an example for making here to decorate the microtubuli, you know. We also have some tubuli which have a specific function, like in our cilia. Think about, for example, the best cilia that ones, you know, are the ones of sperm cells, of course. Which are propelling the sperm head, you know, also 30, 50 centimeter to a different place. But we also have cilia in our kind of lung epithelia, and where they make a unidirectional stroke always to move out every kind of dust and whatsoever out of our lung. So these are actually pretty complicated here, because now we have not a single microtubulus. We have now here 9 times 2 microtubuli, which are fused here. Here, 9 times, and we have a central pair of microtubuli. And they are cross-linked by these radial spokes here. And in between, we have the motor proteins, dynein arms. So this is pretty large. You see that here we are already in the micrometer range. So here in coming almost out of the electron microscopic sight range for this molecular machine that is coming up with this organization. The dynein structure is very, very complicated. It has been recently solved by cryo-EMs. There's some major parts of this one here. I haven't put an actual slide of this one in here. Very complex. We will not go in this. The hard part is the ATPase domain again. In this case, the ATPase is forming a hexameric complex. We find hexameric ATPases also in some other protein components, where actually you have to make a molecular movement. So AAA ATPase is a kind of abbreviation for this kind of hexameric ATPases. They also occur, for example, in the machinery for protein degradation, in the ERAT machinery, in the plasmatic reticulum. They also do occur in the kind of vesicle fusion machinery. And there's NSF is also also using a hexameric ATPase domain like that for transporting, making conformational structural changes. In case of the dyna of the cilia, that looks a little bit like this. I took this one from the Source Molecular Machines, probably website. Yes, you see that's actually what it's doing. The dyneins are not here; they keep strongly as they, but they change their relative structure to each other. The nexons are just flexible linkers here, so they move upward, backward, and by this, the cilium makes then this kind of stroking movement. So it's not rotating; it makes this kind of stroking movement that propels forward, like when you swim. Also, a bit, propels forward the cell attached to it, or the kind of things given. Please, some more people of you like to work with bacteria. We have here a lot of bacterial interested people. Please remember, we have tubulins also in bacteria. Of course, also then in here as well in some way. The most famous one is FTSZ. FTSZ, this protein looks very similar, like we have seen the tubulin before, or the kinesins before, this looks a little bit like a mixture of both, yes, but actually it is forming ring-like structures. This protein is making ring-like structures, and when activated, it can start to make movements of these rings relative to each other and can start to shrink them. So it's very important for making rings of the division septum, where the cells start to divide and being separated, the bacterial cells. So this is apparently an evolutionary very old family, a prerequisite for cell division. We also have them here in gut epithelia. I just flip over this. Actually, there's something in German written, Let's go to F-actin or to actin. F-actin is the polymerized form of globular actin, G-actin. Architecture is a bit simpler because we only have here now, let's say, something like 2 protofilaments which are forming this kind of F-actin. You see this one and the other one. They are intertwined with each other. So here are some of the geometric parameters, but you can read by yourself. Nevertheless, they have a plus end and they have a minus end. So you can keep up in mind, they are also dynamic, plus and minus end. So we have, similar like with the tubulins, also a cycle. So in this case, we have it a bit different. So the monomeric form is the globular actin is the ATP occupied form. So it's exchanging ATP with ATP and vice versa. It's in the in the globular form. Upon hydrolysis of ATP to ADP and phosphate, it starts to polymerize. It forms the fibers. So this is simpler a bit, yes, than with our friends tubulins. Actually, it's the other way around, you could say. There we use GTP, here we use ATP. In that case, GTP was driving polymerization. Here in this case, it's ADP that is starting to polymerize, just the other way around. Mechanism nevertheless similar, yes, we have here this kind of filaments, they're also dynamically unstable. We have here a 4-domain architecture with the ATPase domain attached to it, and the other domains, acting, for making the kind of interactions with the subsequent preceding and subsequent actin molecules in it. They also change the conformation or the relative orientation upon hydrolysis of this ATP here. You see this here as part of this kind of fiber. Pretty nice structure. Also relatively simple, in terms of the molecular details, because again, we have a P-loop region, something like a switch region, something similar, and also causing then some conformation. So you see that's the difference between the two structures, between the ATP bound form and the ADP bound form. It's not a very large structural difference, you can say, yes, it's just a minor rearrangement happening here, but that's enough already to change the oligomerization state. So we have, of course, models of actin fibers being done pretty early. Nowadays, one has also experimental structures of these kind of fibers available, not only this kind of first approaches where one had just a few molecules co-crystallized to each other and was happy to get a structure. One can get now with also electron microscopy or X-ray diffraction. That depends a bit, also from fibers, diffraction patterns. Fiber is something like a one-dimensional crystal. A one-dimensional crystal with its long distance regularity, its translation symmetry is causing diffraction, so that one can actually get some geometric parameters from the diffraction patterns, like you have it in normal protein X-ray crystallography, where you have three-dimensional crystals, or in electron crystallography, where you have two-dimensional crystals. So when you do this kind of fiber diffraction, you also get your kind of diffraction spots, which can be all interpreted, and which give you then the distances. And together with the kind of phases which you can measure when you use electron diffraction, not X-ray diffraction, but electron diffraction, where you also get some phases, you can calculate also the kind of envelopes, of these fibers. And you see this is done here, for this kind of fiber, for this F-actin fiber, and here is in colored, the kind of monomeric actins have been just puzzled in and seen they fit almost perfectly into this low resolution structure. I guess the resolution is in the way of about 15 to 25 angstroms, so something where we do not see structural details, at least at this stage. I guess there will be some advancement of getting better structures of F-actin fibers. So just to remind you, finally, about this kind of dynamic proteins. They are not finished. They co-occur in the cell. All three of them. The intermediate filaments, okay, they're not dynamic, but the tropomyosin here shown in green, and the actin, it's colored by phalloidine, here shown in red. And you see here, for example, it's making here a lot of some stress fibers. They are mostly made of actin, and also here of this kind of tubulins, which are also coming up here to form the structure dynamic. Probably this fibroblast is migrating over its ground now. It's making now some filopodial structure here and moves into that direction. So here starts polymerization, and at the opposing side you can predict that's the side where the depolymerization is happening. So this is a highly controlled process, especially of actin, because it's forming these kind of stress fibers. There are numerous numbers of actin interacting proteins. I will not give you some conclusive overview. I think that would be something for us cell biology friends. So we have proteins which disrupt our actin fiber into pieces, severing proteins. We have some which make bundling, like in the filopodia, where we have this movement on, yes. We have some capping, yes. We have cross-linking. We need cross-linking to get some stable structure of our cells, yes. We have something to nucleate, something to depolymerize, something that binds to the site, and also something, that makes motor movements, which I come later about this, because this is something for basic knowledge of structural biology, how actually our muscles do work. So, for example, blocking actin polymerization. You see that here, how this kind of polymerization is blocked. You have a very small protein, this one here, WH2. In the free form, it's probably also completely unstructured. In the associated, it's formed a small helical fragment here, and a long region that is wrapping around the globular actin and just blocking the interaction site with other actins, so it cannot polymerize anymore. Very simple mechanism, just steric hindrance. Here thymosin, so that one here was binding at the plus end. Thymosin binds at the minus end. It also blocks polymerization. Of course, it also blocks depolymerization in that case. The thymosin, from the other side. Very similar, just the helix coming from the other side, and the rest is just wrapping around. Primitive, very primitive way. So you see, that's minimal principle. You want to make something, you use just what is essential. We can, of course, make it much more complicated. You take a real protein, like this one shown here in red, profilin. It also blocks the polymerization. Now we have a really structured protein, yes, and that can become part of larger machinery as a profilin. Now everything becomes more complicated, Here we have gelsulin, yes, that's also blocking the surface. Now we have a 6-domain protein that actually can be also regulated by interaction with some other proteins. In this case also calcium. There's some calcium binding sites. So calcium, as we have seen it here, calcium can bind to some aspartates, disrupt the salt bridge there, and triggering it, for being for having affinity to the actin and to bind to the end to this. So that's a calcium triggered capping of the actin fiber. So calcium can be then used, influx of calcium in the cell to steer it. Another one is ARP2, ARP3. We see this here, it's forming with an adapter protein a branching end, yes. So where we can branch out our actins and make some side branching. Very nice, very complex, yes, I like this super complicated structure. You see, now everything becomes more complicated. More and more building blocks to come in for this kind of mostly structural purposes. So that's really like Lego bricks. Lego bricks made on a molecular level where you combine all these kind of components. And actually that's just an evolution by us. Here's an open conformation of ARP3. ARP3 is binding also a nucleotide. Actually, I think it's GTP that is bound here, and then it's closing in, and getting also functional again. We have actin homologs in bacteria as well, MIEB, for example, in bacilli, like Bacillus subtilis, on which Peter Kahorman is working. It's responsible for making these two rods, yes, It's not cocky, yes, but rod-like. Some of the shape-forming parts of this kind of bacteria. Now at the end of today's talk about our motor proteins, I take the muscle motor, yes, our muscle motor, yes, of all kind of animals, yes, the fantastic motor, yes, especially it's a bit more advanced animals. So the contractibility, that's not the unusual thing. I think almost all eukaryotic cells can in some way or the other contract, I think that's mostly done by the actin network. In humans, we have different types of muscles: skeleton muscle, smooth muscle, hard muscle, 3 types, 3 major types. There's, of course, subclassification of the muscle fiber types in skeleton muscles, fast and slow fibers, but that doesn't matter. We see here this explosion, it's a bundle of muscle fibers, or muscle, it's a multicellular, it's a multinuclear cell actually, which is very interesting. A lot of muscle cells have fused to giant cells, which have a lot of nuclei, where each nucleus is responsible for a certain kind of part, of this giant cells, which are formed upon cell fusion. The myofibrils are crossing these kind of cells, and they are highly structured, as can be seen in electron microscopy, into this kind of band-like structures. I band, A band, and the central disk, and the M line, and from one Z disk to another one that defines one sarcomere, and we have a lot of sarcomeres, and actually almost one nucleus per sarcomere. And that's actually the overall architecture. And this is enwrapped with the sarcoplasmic reticulum, which is just an endoplasmatic reticulum here specialized for this kind of polynuclear cell. So when we make a cross-section to muscle cell, you see it's highly organized, the kind of myofibrils in it. This is here, that looks like a crystal. It's a crystalline-like arrangement, yes. And each one of them, is comprising myosin fibers and our actin fibers, So this is a pseudocrystalline arrangement that we do have here. So in charge, making this movement. Like with the cilia that we have seen before, where we make the movement of our cilia, on the cell of the sperm cells or lung epithelial cells, which also makes a movement. But now this is now blown up, in some macroscopic structure. So here, just to remind you about this kind of different kind of nomenclature, it's the EM image, negatively stained, of course. A lot of stain is found in the Z disk, yes, a bit of stain here, and here this is protein rich. Here is something actually where we had the cross section before. That was the pseudocrystalline arrangement here in in this part of the A band. So let's make a look, what's actually to be found there. The general architecture of this kind of motor setup is defined by the Z disks, and between the Z disks we have a span of titin, which is a rubber-like super large protein. Come about that later. That is some of the framework. Attached to the Z disks are the actin fibers. They are attached with their plus ends here at the Z disc and with the minus ends close to the M line. So these are also fixed. What's not so much fixed is that part here of the myosin. That is part here. The myosin is here also attached to the titin, but it's occupying the central positions. So when you compare that, it's really like an apparatus, with the central part which probably can slide, yes, inward. See, are gigantic proteins, nebulins, 7,000 amino acids. Titin has 34,000 amino acids, Super large proteins to make this kind of architecture. So titin actually is a very interesting protein. It has a molecular mass of 3 million Dalton, much more mass than a ribosome. Cool. There's 320 domains. Clearly, each domain has about 10 kilodalton. Of these 320 domains, about 41 belong to a domain type that is very familiar to us. It's the immunoglobulin type that we have found, for example, in our cell surface receptors. I think when we talked about our receptor tyrosine kinases, they are found, of course, in our antibodies. They are found here, 41 copies at least of them are found in titin. And what's cool about it, why it's acting as a rubber band, it's not only folding domain-wise by domain-wise, but the domain can fold and unfold. So each time when I stretch and compress the muscles, I fold and unfold this titin that is part of this machinery, each immunoglobulin-like domains. That goes really on-site reversible. So protein folding and unfolding here just to have this kind of rubber band-like consistency, so that to restore the shape again. That's actually what the rubber band is doing. It has some elasticity, try to reshape it. In this case, it's a folding energy that is driving it forward. So that's about more with the architecture. You can look at the textbook, let's go for our myosin actin system, the sliding filament hypothesis or theory, unfortunately this is one of the remaining slides in German, yes. So the filaments themselves, they don't change the length at all during the contraction. So they just glide along each other. So it keeps constant. That also means that the length of the eye filaments of the eye region, this region here, keeps constant, where they are, and also the A region keeps constant. The A filaments, they are assigned to the name for this. The miracle is in the heads of the myosin. We come about this later. Here, just about this idea, everything keeps constant and moves forward, backward. Here, that when you eat your steak, yes, 90% of what you're eating of your steak, yes, is myosin and actin, nothing else, yes, a bit of titin and nebulin and other things. But that's the reason why we probably like beef so much. It's just almost pure protein, yes, It's almost pure protein diet. It's fantastic. So the speed about 8 micrometer per second per sarcomere. But when I make this kind of movement, I move much more, of course, yes, or my muscles move more because there are many sarcomeres behind, yes, lined up. So myosin has also this structure that we have seen in some ways a little bit reminding us of what we have seen with the kinesins, They have here the carboxythermal tail that is forming this long dimers, yes, This is a coil helical region. This is a tail. It's about 15 micrometer long, 0.15 micrometer long. Sorry for getting it wrong. So it's pretty long, 0.15 micrometer. These are the heads, which are about 17 nanometer long, which is pretty large nevertheless. So it's a protein complex here, you know, it's these heads, you know, which comprising light chains, 2 different types of light chains, and the heavy chain is actually that one shown in red or in light red with each other. So that's myosin 2, one of the major components. So these kind of myosins are then also forming aggregates with each other. You see that they are bipolar, so they have no different ends, so they are symmetric. So they associate with each other here with the heads, so form like a spiral here, you know, around it with the heads, and in the other way as well. Here's a common association site and a lot of lateral interaction of this kind of myosin dimers. So the basic unit is always dimeric stuff. So when seeing about the kind of components, clearly in the past biochemists used proteases to dissect very large complexes. You see the kind of nomenclature comes from the treatment with some simple proteins like trypsin and papain. So that's what we know. We have structures here of the heavy meromycin, especially of the head groups of fragments of it. Clearly the complete structures of complete myosin oligomer. That would be of course cool, you know, but it's very difficult to assemble. So let's see what the head groups are looking like. Here's chicken myosin, the S1 fragment, one of the very fancy structures, you know. You see the actin binding site, here's the central beta sheet. We also have here the ATP binding site. I don't see it, should be somewhere here around, you know, in this kind of actin binding site, you know, and then the attachment to the rest of the filament here. So that's actually where the myosin head is actually coming in, you know. The actin filament is not smooth, you know, it has this kind of grooves, and in this kind of grooves, the head can actually bind to it in an ATP-dependent manner. But that is not so trivial, because, you know, this groove is not forming by itself. It's formed by having the actin filament associated with a regulatory filamentous protein complex. The called thin filament. So you see here the F-actin and its G-actin components, and we see here the tropomyosin and the troponin complex being associated to it. The tropomyosin is somehow entangled around it. It's a coiled coil forming here, you know. And the troponin C complex, that is interesting because that is the regulatory part. The troponin C complex, the troponin C complex comprised of C, I, and T. C is the interesting part because that is the protein that looks like calmodulin. It has the same architecture as calmodulin, binds calcium and changes its tertiary structure. And this change in the tertiary structure of troponin C is transmitted via troponin I and T to the tropomyosin. So first to the troponin complex, here we have C, I, and T. Here is the calcium binding site 1, 2, 3, and I think this is the fourth calcium binding site. You know, that is a complex. It's somehow reminding the hand that is holding here the chopsticks of made. One is troponin T and the other one is troponin I. Yes, you know, the chopsticks that are making the interaction with the tropomyosin component that we have before. And just to get it, you know, here the calmodulin looks really like our troponin C, and you see also, you know, actually we have it also on the myosin head group. Also, the myosin head group also has a regulatory light chain and an essential light chain. They are also of this calmodulin type, so that's super cool. Yes, you know, everything is here made by this simple protein to be regulatable by calcium. The binding site of calcium is super simple. I think we already have seen it in one of the introductory lectures. It's an EF hand. EF because people called the one helix E and the other helix they called F. They were not very fancy, and these 3 fingers is a short beta strand, yes, you know, that is donating some carbonyl groups for coordinating the calcium ion here in the center of our, it looks like IS, yes, I know. It's a political symbol that should not be shown here in this context. Okay, but you know, that was existing before they came up with, yes, you know. So I will not go about this, also the kind of multitude, we have more than these myosins. There are a lot of myosin-associated proteins, which are intact, also with the actin, everything is super complex, yes. Myosin 1 and 2, we talked about myosin 2, of course, but there's also myosin 1 existing, and as you can imagine, myosin 1 is also a motor protein for moving some cargo on actin fibers, which is probably also the origin of this kind of apparatus for our striated and smooth muscle apparatus. Originally, this kind of myosin 1, the related protein, has been simply a motor protein to transport something along an actin fiber, and only later more complex organization forms have been formed then this kind of actomyosin complexes that we do have in our muscle. Now let's see whether we have also some fancy movie. I like to have now the fancy movie. That is a fancy, simple one, yes. You see here our myosin, yes, myosin complex, head group of myosin. We see here in purple our troponin complex with the C, I, and T. Remember this chopstick idea, yes, And each time the calcium binds to it, it moves the tropomyosin to the side, so that actually the head of our myosin can actually touch the actin now. And when it's touching, you see that those are here shown schematically, yes, it can start to bind, hydrolyze ATP, gets being released, and make one stroke. So that is, these are all the components that we do have. So we have different structures of the myosin head group. Again, we don't have them in complex with ATP, because ATP would be, of course, hydrolyzed by myosin. Again, we use a chemical trick. The chemical trick is to use some analog of ATP, and that it can be done by using the ADP aluminium tetrafluoride complex. Aluminium tetrafluoride is an anion. I hope that you can follow this, that it's a single negatively charged, and is associated because it's more or less planar, coordinated, the central aluminum ion and the four fluoride atoms. It can associate with the terminal oxygen of the ADP, that is from the beta phosphate. So here ADP and here aluminum, that's coordinated, and then our fluoride ligands that we do have here. It's forming a nice complex. Interesting, this complex is only formed not in free solution, but only in ATP binding site. So it's formed in situ. And the same trick works also with beryllium trifluoride, which is also negatively charged, single negative charge, as you should remember, yes. It's also forming a very nice complex between this oxygen coming from the ADP and the beryllium atom. So that's a way to make ATP analogs. The other way would be, of course, to make a non-hydrolyzable ATP analog by replacing the oxygen by an NH imide group or a CH2 group, methylene group. So we have it here now in the ATP bound-like form existing, So that's pretty close, And that is the ADP bound form. And you see here, it's like with the kinesins, it's releasing this kind, like we have seen it with the helical neck region. It's releasing the C-terminus. It's a similar way, a bit different than with the relay helix that we have seen, The architecture is a bit different of the myosin, but the principle is always the same. Post-trigger chicken skeletal muscle. And that's when the myosin is empty and has discharged the ADP, then it's completely open. And that's again, I think, our movie, yes, that we have here. It's binding. Phosphate gets out. ADP gets out. No. ADP gets in. Now it gets off. By hydrolysis, it gets off. And then the same thing again, yes, The ATP gets in, hydrolyzed, it gets off. So that's a different way, how that goes, So the dissociation occurs by the ATP hydrolysis, yes, So here, ADP bound form, ATP gets in, Actually, ATP hydrolysis drives it off and also makes a conformational change, from back to forward stroke. Yes, backstroke, forward stroke, hydrolysis. Then it binds again. Then actually it releases phosphate. Then it waits again to exchange with the ATP and the cycle repeats. That is actually a repetitive cycle. Almost very similar like we have seen it with the kinesiones. At least, not from the very, very details, but from the principle, always very similar. Vanadate is actually the fourth option, how to make an ATP-like complex. Use vanadate, yes, which is also forming in situ a nice complex in ATP binding site with ADP. So when you are in search for an ATP-bound structure, please remember there are 4 different ways to make the structure. Good, that's about this myosin stuff. We have here also a relay-like helix. Yes, they also called it now relay-like helix. It's not taken from the stria and switch 1, switch 2. It's pretty similar. Also the details are a bit different, yes, and it's from the other textbook, yes. One thing that I think of interest when you have a motor, a molecular machine, what about this kind of transmission? So interestingly, you have an influence, of course, of velocity on the length of the lever. Yes, that means by which actually it changes with this arm, the changing, whether it's this way, that way, or that way. So that is a very nice demonstration. Yes, that the number of light chain binding sites and the sliding velocity, and these are the chains here, and you can fuse it, then that one varies it, can have an effect on the sliding velocity. Clearly, you have to keep in mind, that you have an angular momentum and that you make a certain kind of amount of work, and that will be, of course, not changed at all. Yeah, so it means, that actually when you have a longer lever, of course, the kind of force that can be then applied to something is not necessarily becoming bigger, because the way over which the force is integrated, it's the energy, has to become smaller because of the longer parts that you are actually using. But this is possible. That is one of the things, also something to evolutionary optimization. Then in smooth muscles, it's the same, yes, I don't show it in smooth muscles. The myosin superfamily, 17 subfamilies, super complicated. This is the unrooted phylogenetic tree from 2000. I don't know how it looks like nowadays. I have to look for a new one, I guess. Maybe they have now 18 subfamilies now found, or 19 with something. So it's a very ancient family, that's at the beginning of all animal life. We also find it, however, also in other organisms, not only in animals. We have myosins present in almost all eukaryotes. So, for example, there are also some plantal myosins here, this green family. Yes, plants also make some movements. Don't forget this. Can be also motor protein. Some of these proteins are motor proteins. We find them also in some single cellular organisms, like, for example, Pantoffeltierchen in German, or Tetrahymena, I think, in the systematic language. Okay, so maybe we finish now for today. I think, what do I do to finish? I take this educational video from McGraw-Hill and let it run. You use muscles every day to do activities. How long actually is this movie? Oh, it's about 4 minutes now. We can look now for this. Yes, blah, blah, blah. I make it a bit faster up here. Sarcomere. You can look it by yourself, yes, in YouTube, no problem. Very nice, very, very nice animations to see them happening. Yes, thin and thick filament, we talked about that. Upsala, it finished already. No, I'm already at the end. That was a very quick end, you see, this time. Thanks a lot for your attention, and we see each other next week. Yes, don't forget, we have Thursday our first seminar date. Bye-bye.